Okay, we are the Wing Lab, and this is our presentation on engineering bi/trispecific antibodies to target acute myeloid leukemia. Um, we'll first go over some background and context, and then we'll move on to results, our methods, and yeah. So firstly, some background. So what is acute myeloid leukemia? Myeloid leukemia, or AML, is a blood and bone marrow cancer that is most commonly diagnosed in adult patients. AML can develop from myeloid stem cells and myeloid blast cells, making the bone marrow produce abnormal cancerous white blood cells that deprive healthy, non-cancerous blood cells of nutrition. Because AML is a blood cancer, it metastasizes and progresses quickly, using the bloodstream as a method of transportation to easily infect nearby nervous and lymphatic tissue, which contributes to its low survival rate. The 5-year survival rate of adults is just under 30%. Its diagnosis is relatively simple compared to other types of cancer, as it requires a blood test, which is both non-invasive and low cost. Chemotherapy is still one of the most common treatments for AML. FLT3 inhibitor drugs only target variants of AML with mutations in the FLT3 gene, and this makes this more targeted than chemotherapy. IDH inhibitors only target AML variants that have IDH1 or IDH2 mutants, which affect blood cell maturation. The IDH inhibitors block mutant IDH enzyme activity and restore normal blood cell maturation. Anti-CD33 ADC is used mostly in limited cases and is an antibody which is conjugated with a chemotherapy drug. This antibody directly binds to the CD33 protein, which are mostly present on the surface of the leukemia cells. This allows the chemotherapy to be delivered more precisely. CAR T cell therapy works by genetically modifying a patient's T cells to target AML cells and is still in development along with many other bispecific antibodies.
So now we'll be talking about the bispecific versus other therapies. Bispecific is a better therapy because its effects extend beyond when the cancer drugs are in the body, as the body itself is taught to fight off cancers. It only kills harmful cells, not beneficial cells, and the side effects are generally not as harsh. Other methods, such as radiation therapy, may damage DNA and organelles, while tumor cells can avoid being killed. Surgery is invasive and high risk, while chemotherapy uses large quantities of drugs to kill cancer cells, and it kills both cancerous and beneficial cells, and its effects only last while the drugs are in the body. Now, what are antibodies? Antibodies are Y-shaped proteins essential for immune defense, consisting of 2 heavy chains and 2 light chains. The Fc region, located at the base of the Y, binds to accessory cells and is consistent across antibodies of the same class. The Fab region, located at the tips of the Y, are variable and target different antigens. While antibodies themselves do not directly recruit T cells, they play a crucial role in marking antigens for destruction and facilitating immune responses, which can lead to the activation of T cells through antigen presentation. And now we'll be talking about the bispecific approach.
So how does the bispecific antibody work in our project? The bispecific antibody is unique because it has 2 fragment antibody binding regions called FABs instead of 1. For AML in particular, one arm binds to tumor antigen protein CLL-1 or CD33. These are cytokine receptors that are highly and differentially expressed in AML blasts compared to normal healthy tissue found elsewhere in the bone marrow. The other arm of the bispecific antibody binds to the T cell engager CD3. This engagement is crucial as it activates the T cells, which are immune cells responsible for killing cancerous and mutated cells in the body. The end result of the bispecific antibody binding process is simple: the eradication of the tumor through T cell proliferation, signaling, and the redirection of cytotoxic response. On this slide, you can see that there are some possible target receptors for bispecific antibodies treating AML. These receptors shown here are characterized by being differentially and overly expressed on acute myeloid cells to prevent off-target effects from damaging healthy tissue found elsewhere. In our project, we are specifically targeting the TIM-3, CD33, and CLL-1 receptors. As mentioned previously, CD33 and CLL-1 are both cell surface markers found on AML blast cells, and TIM-3 is a self-surface protein found on leukemic stem cells and works to suppress immune cell responses.
This is the design of our bispecific antibody targeting CLL-1. On one side, we have an anti-CLL VHH domain, which specifically binds to CLL-1's receptor that is overexpressed on AML blast cells. On the other side, we have an anti-CD3 domain, which engages T cells. These two regions are connected by linker, allowing the antibody to bring T cells into direct contact with leukemia cells. This is our anti-TIM-3 bispecific antibody. The orange domains are anti-TIM-3 VHH, connected by a linker in green to an anti-CD3 antibody shown in blue and gray. This design allows the antibody to bind to TIM-3 on tumor or immune cells and CD3 on T cells. And this is our anti-CD33 bispecific antibody. The pink domains are anti-CD3 CD33 VHH connected by linker in green to an anti-CD3 antibody shown in blue and gray. This design allows the antibody to bind to CD33 on a tumor or immune cells and CD3 on T cells. CLL-1 is present on most primary AML cases, ranging from about 77 to 92%. It can serve as a marker for leukemia stem cells, which are thought to be relapse in leukemia. Importantly, it's not expressed on normal CD34 plus and CD34 minus cells. The graph on the right represents this. The AML cells have high CLL-1 expression, while NMB, RBM, and MBP show little to none. This makes CLL-1 useful in distinguishing leukemic stem cells from normal stem cells.
So now let's move on to the methods. The procedure begins with the production of plasmids, which encode the heavy and light chains in E. coli cells, which is then followed by culturing on agar plates and the DNA extraction via midi prep. These plasmids are then introduced into cultured mammalian cells to drive antibody production. After transfection, the protein expression levels are extracted and measured to confirm the sufficient yield. Then the heavy and light chains are subsequently combined to generate functional bispecific antibodies. Finally, their viability is assessed using MTT assays and cytotoxicity is evaluated to ensure their effectiveness in targeting acute myeloid leukemia cells. Next slide, please. So the cell lines we use here are the HL-60 cell lines, which are derived from a patient with acute promyelocytic leukemia. HL-60 cells can differentiate into various mature myeloid lineages, making them a valuable model for studying immune responses. We also use the THP-1 cell, a monocyte cell line isolated from the peripheral blood of an acute monocytic leukemia patient. Under different culture conditions, the THP-1 cells can differentiate into macrophages and dendritic cells.
We use flow cytometry to verify the presence of antibodies in the sample by checking their size and complexity through fluorescence or the excitation of probes. This technique was then employed to analyze the binding ability of these antibodies to leukemia cells, specifically the HL-60 and THP-1 cell lines. Now we're going to talk about our preliminary results. As you can see here, this is our first proof of concept that shows that our bispecific antibody can effectively bind to the tumor cells. We produce these antibodies using VHH1, VHH2, and VHH3 antibodies. These can all bind to the human-sized CLL-1, cynomonkey CLL-1, and mouse CLL-1 antibodies. The cynomonkey CLL-1 is especially important because the antibody must be tested in non-human primates for initial cytotoxicity. These are all kept stable at 37 degrees Celsius for 1 week, and our peaks were generated from flow cytometry, and they show the binding affinities for the different antibodies to leukemia cells. The secondary antibody by itself, which is just the fluorophore, showed a residual binding. On the other hand, CD3 by itself had minimal binding. All three bispecific antibodies displayed relatively high levels of binding, which kind of shows that our cells can viably bind to the tumor cells. This is our second proof of concept that kind of shows that our antibodies can effectively kill the tumors. As you can see, the HL-60 peak is higher for cytotoxicity analysis, which corresponds to the higher peaks in flow cytometry. Anti-CD3 antibodies only bind to the T cells and not specifically to AML, which is why the anti-CLL-1 CD3 is the bispecific antibody that shows much higher binding, even in HL-60 cells. PBS and secondary antibodies are shown as controls to demonstrate the difference in binding capability a lot more clearly. These results have also been confirmed visually with our MPG assay. Our MPG assay results showed a significant decrease in the purple formation intensity in AML cells treated with our bispecific antibodies. This indicates that the antibodies effectively induce cell death. leading to a reduction in viable cells. This positive outcome confirms the efficacy of our engineered antibodies targeting and eliminating AML cells, demonstrating their potential as effective therapeutic agents. Where are you muted?
I can't hear anything.
Where are you muted? Where are you muted?
I'm so sorry. On the left, we have a THP-1 AML cell line, and the right shows THP-1 TIM-3. The top controls, with no antibody and only secondary antibody, show baseline signal, while anti-CD3 and anti-CLL-1 antibodies bind as expected, showing a positive signal. Importantly, when we test our anti-TIM-3 VHH antibodies, we see a clear binding to the TIM-3 expressing THP-1 cells. This confirms that our engineered antibodies are specific and functional against TIM-3.
So we have some preliminary results for testing anti-TIM-3 CD3 bispecific antibodies on THP-1 cells and THP-1 cells enhanced with stable TIM-3. And so the histogram on the right shows a steep increase in cell death at 50 nanomolar, while lower concentrations lower than 0.01 nanomolar give minimal effect, so we can take away that the antibody is dose dependent with a clear potency threshold around the high nanomolar range. And from this, we can conclude that constitutive TIM-3 expression amplifies cytotoxicity, especially at the top concentrations.
Building onto bispecific antibodies, we have the trispecific approach. The aim of bispecific antibodies is to have efficient recognition and targeting, as well as safe discrimination of target cells. When focusing on efficient recognition and targeting, the activity is high and expression is in all tumor cells at a high in many patients. Safe discrimination of target cells has low activity and is not found in any normal tissue. By recognizing two leukemia markers at once, this design increases cancer cell selectivity and reduces immune escape.
So the aim of bispecific antibodies is to achieve both efficient recognition and targeting of tumor cells, as well as safe discrimination from healthy cells. So our trispecific antibody combines three main binding regions, which are the anti-CLL-1, anti-TIM-3, and anti-CD3. And the trispecific antibodies modulate their activity based on which markers are present on the cell. So if a cell expresses CLL-1 but not TIM-3, as shown on the left, the trispecific antibody can still bind through the CLL-1 arm and recruit T cells, so it's still able to kill, but the potency is much lower compared to double positive tumor cells. And in the middle, if a cell lacks CLL-1 but it still expresses TIM-3, the antibody is unable to kill. And this is important because that is the typical structure of a healthy cell, and we want to avoid killing healthy cells as much as possible. And finally, on the right, when a cell is double positive for both CLL-1 and TIM-3, which is a characteristic of AML cells, the trispecific antibody shows the strongest effect with the highest relative killing capacity and potency. So this mechanism allows for the selective and concentrated killing of leukemia cells while minimizing damage to healthy cells. So with the trispecific design, the antibody adjusts its activity depending on the type of cell it encounters. So for normal cells or cells that only express one of the target markers, toxicity is lower, and this reduces off-target damage and patient side effects. But when the antibody binds to double positive tumor cells, which are far more characteristic of AML cells, the response is much stronger and much more potent. And targeting multiple markers enhances tumor selectivity and lowers the chance of immune escape, since the cancer can't simply lose an antigen to avoid detection. And with the third arm, the antibody directly recruits and activates T cells at the tumor site, boosting their ability to recognize and kill cancer cells efficiently, which in turn leads to a stronger and more sustained immune response. And this slide shows some possible target receptors for trispecific antibodies treating AML.
So here we have some results testing the trispecific antibodies on CLL-1 low and TIM-3 low cells and CLL-1 low and TIM-3 high cells, which show that trispecific antibodies enhanced leukemia cell killing on THP-1 TIM-3 expressing cells. And as you can see on the graph on the right, which is TIM-3 high, there are sharp death peaks at around 0.08 nanomolar and 0.04 nanomolar, while on the left graph, on the TIM-3 low graph, it remains relatively flat. And from this, we can draw that TIM-3 provides a critical kill signal when CLL-1 is scarce. And on this one, we have a CLL-1 high and TIM-3 low cell. So the bar graph compares anti-CD3, which is the baseline, to bispecific antibodies, as well as the trispecific antibodies, which are the ones in magenta and pink. And the trispecific antibodies achieve the highest death percentages across the full concentration range, outperforming both the bispecific and monospecific antibodies. On this graph, we have the results for CLL-1 high and TIM-3 high cells, and it shows us that when CLL-1 is highly expressed, adding TIM-3 does not further increase killing, which implies that the TIM-3 benefit is context dependent.
So now we'll be going over some future steps in our project. Next slide, please. In terms of future steps, we hope to conduct in vivo assays to test AML binding affinity and cytotoxic activity. During this step, we would also ensure that no off-target effects are present. If our testing proves successful, we plan to apply the same bispecific method to ovarian cancer, including antibody generation, flow cytometry, MTT assays, and in vivo analysis. And finally, we want to continue our work on developing the trispecific antibody, especially for TIM-3 expressing leukemic cells.
I would like to give a special thank you to Dr. John Wang for the countless hours of valuable guidance, mentorship, and support he's given us for this project. We are also immensely grateful to ASTRP for providing us with the opportunity and workspace to do this research. Lastly, a quick thank you to our parents for chauffeuring us around and being patient with us throughout this process.
Any questions?